Hey everyone, Dr. Jack Ordy, and in this video we're going to discuss how to use modern techniques to actually figure out how drugs work. Now in the previous video I explained that it is actually really difficult to figure out how drugs work. There was, for example, there was 130 years between the invention of paracetamol, acetaminophen if you're in America, and the discovery of how paracetamol actually works. It's a very difficult thing to do. Now in this example we're looking at how do antibiotics work. How do we figure out how an antibiotic is able to selectively kill bacteria without killing our own cells? And in this really clever example, we're looking at how to use the power of evolution to figure out how antibiotics work. So let's jump into it. So first up, there are lots of techniques. There are pull downs, computer modeling, genetic knockouts, RNA sequencing. All of these I discussed in the previous video. But in this one, we're looking at resistance evolution, i.e. the evolution of bacteria to become resistant to your drugs, combined with next generation DNA sequencing. So we're trying to use the power of evolution and the development of resistance to your antibiotics that you've just invented to figure out how those antibiotics actually manage to kill the bacteria without killing your host cells. So let's jump into it. There's a great paper that breaks down this technique called Toprak 2012. Really, really good paper in Nature Genetics. And in it, they discuss this device, which I love the name of the device. It's so good. It's called the morbidostat. And essentially what it does, let's have a look at this diagram. We've got over here is where the bacteria are being grown. They're being grown in this chamber here, and they're going to be stirred by a centrifuge, and they're just growing in a suspension in a nice brothy media with all the nutrients that the bacteria needs to grow. Now over here, we've got this thing called light absorbance, where basically you can shoot a laser through your culture, and based on how much of that laser can get out the other side, which we call optical density, it tells you how many bacteria are in that flask. Essentially, we're looking at how cloudy the solution is. You shoot 600 nanometer wavelength through, and how much can get out the other end tells you how cloudy the media is, and how cloudy the media is tells you how many bacteria there are. Now, this is actually set up to a decision computer, probably an Arduino or something. It's a very simple yes-no question, and probably in a computer chip that says, if the density gets above a certain level, we're going to open this valve, and this contains the growth medium plus drug. And so what it will do is it will add that drug in, and that will contain some level of antibiotics, normally a low level of antibiotics. And if there isn't a lot of bacteria, every hour it might just add the regular medium. So every hour it's going to evaluate the optical density and decide whether to put in regular growth medium or regular growth medium with drug. Now what's going to happen is the goal of this device is to maintain roughly the same number of bacteria in this flask. And if the bacteria get to a very high level, it's now going to add a little bit of your antibiotics. Now what that's going to do is kill off some, but not all of your bacteria. And then what happens is the ones that manage to survive that low dose of antibiotics will grow. And then if it gets to a certain level, they'll be administered antibiotics again. So why do this? Why develop a device that will administer antibiotics to maintain a stable population of bacteria? Let's have a look at what might happen. So here we have time, and here we have bacteria number, also optical density. That's what OD stands for. So that's how much of the light is absorbed as you shoot it through the culture medium. So this is what we might expect. So you seed the bacteria, they grow exponentially as bacteria do, and then they start to curve off as they crowd out their environment. So you're sort of getting to the maximum capacity of the flask. And then what's going to happen is you're going to add your antibiotics, and that's going to kill a lot of the bacteria off. And then they're going to grow. And then you're going to add a higher concentration. So here we can see you start off with a low concentration of your drug, and then you get higher and higher and higher. And so eventually the bacteria will become tolerant, completely tolerant and resistant to your antibiotics. And largely this is because of evolution. So the bacteria here that survived this low dose of antibiotics must have some sort of genetic variant that allows them to be a little bit resistant to this concentration of drug. They will then survive and grow, and then you can administer a higher concentration of the drug and they'll survive and grow. And what we will do is we will accumulate mutations that improve the survival in higher and higher concentrations of antibiotics. It's essentially the worst way to treat a human being, right? If you have a staph infection, we don't want to give you a low dose of antibiotics to ensure a good number of those staph survive and then increase the dose. We want to give you maximum dose to wipe out the antibiotics. Make sure you complete your course, although there's a bit of interesting research around that. So now doctors have shrunk the courses because it turned out too long a course is a bad idea. But anyway, you want to hit them hard and early so none of the bacteria survive, rather than trying to gradually increase the concentration of antibiotics, because the mutations and resistance will evolve and develop over time. So by the time you get to here, you have a culture of bacteria that have survived an incredibly high concentration of your brand new antibiotic, and you don't know how your antibiotic works, but now you know you've got bacteria that are resistant to it. Now you don't want those bacteria to get out in the wild, so you've got to be very careful. You've got to sterilize everything and all that kind of stuff. So here's a picture from the actual paper. Now in green, we see this drop down. That's just the machine adding the regular media. So it's just adding the regular growth media in this case. And in the purple, it's adding the growth media plus the antibiotics. So we've got 2 doses of antibiotics here. And then because it starts to curve down, they leave it for a wee while. And then a few days later, they start to add antibiotics. So it's actually over quite a long period of time. They add these antibiotics to allow the mutations to occur that provide resistance to the antibiotic. So what do you do next now that you've done this? Well, in all good experiments, you run a control group in which you don't give any antibiotics to, and then you run several replicates in which, so you'd have several morbidostats, that funky machine that administers antibiotics. And so in order to repeat your work, you can't just do n equals 1, you're going to have several replicates. So you have your control groups, and then what you do is you sequence them. Now your 3 control groups will likely be very genetically similar because there was no selection pressure. They just grew and grew and grew. They did absolutely fine. So your 3 or 5 control groups, depending on how many you run, their genetic sequence will be very similar. So here we have our control groups, and they've all got a very similar genetic sequence. Then what you do is you sequence the ones that receive the antibiotics, right? You do a full genome sequencing using next generation sequencing to look at the DNA sequence of your resistant antibiotics. So let's see what happens. Well, largely they'll match. They'll be the same as the control, but hold on a minute, we've got a mutation here. So here in a certain location in the genome, in the bacteria, there is a mutation. And let's have a look at our other replicates. Now, often there'll be a mutation in a similar location or an identical location. So here's our 3 replicates, and what do you know? They've all got mutations in one specific location in their genome. And what you can then do is ask, wait, what is that gene? Now, you might be seeing how this is going to work. They've evolved resistance to your antibiotic, and they all contain similar mutations in a similar gene. So now let's look at the function of that gene and see if we can figure out if it gives an indication on that's how the antibiotic might be working. So in the paper, they looked at this antibiotic called trimethoprim, which we call TMP for short. And what we found was all the mutations, most of the mutations occurred in this gene here, in this promoter region of this gene. The gene is called THFR. It's an enzyme involved in folate metabolism. And I'll jump into that in a second. So N1 had these mutations, the first replicate that they did. N2 had almost an identical set of mutations. N3 had a similar set of mutations and some new ones as well. N4 had some mutations all throughout this gene, and N5 had mutations. So all of these replicates had mutations in either the promoter region or the actual gene itself of this gene, the DHFR. So what is and now let's quantify the overall mutation rate. And what we can see is in the TMP treated bacteria, so this is the TMP treated bacteria, the mutation rate was significantly higher in the DHFR gene compared to all other genes. There was, low levels of mutations in the control group and low levels of TMP in the other genes, but the vast majority of mutations were occurring in this one gene, the DHFR gene. that indicates that that might be the gene in which the antibiotic is working. How do we know this? Well, let's have a look, right? So this is what the DHFR gene does. It is an enzyme, so it catalyzes reactions, and actually it catalyzes the metabolism of folate or folic acid, a very important vitamin that you should take, B vitamin. And folate is important because it's a precursor to a lot of things. We like to modify molecules, and folate is an important thing that allows us to modify other molecules. It allows us to add a methyl group, to be specific. So it actually is critical in the thymidine production and thymine, the T symbol in your DNA, right? So it's absolutely critical in DNA nucleotide synthesis, and it's also critical in the production of methionine, a very important amino acid, and there's many other roles. So you can see that blocking this production is really going to affect the bacteria massively. It's not going to be able to divide because it doesn't have any nucleotides or functional methionine protein, for example, amino acids for its proteins. Now, humans actually have this gene too, but it seems to have evolved completely separately, so it has a very different structure. And that's really important for selective toxicity. The bacterial's enzyme is a completely different structure to our enzyme, and so the antibiotic is able to work on the bacterial protein, but not our protein because of the structural differences. So it's a critical enzyme for bacteria, and the TMB fits into its active site and deactivates it. So this is the antibiotic here in blue. It's slotted perfectly into the enzyme, blocking the enzyme from doing its job. So now it cannot do this critical metabolism of folate. Super important, right? So now let's look at the mutant. The mutant version of this gene has a different shape. It's got a slightly different active site. And so now the antibiotic is incapable of entering the active site to inhibit it. So now we have a functional gene that's going to work. It's a different shape, so now the antibiotic doesn't work. And this is how we can use evolution to figure out how something works, right? If all the mutations are occurring in one gene, and that gene has some important role in metabolism, we can start to infer that that antibiotic must somehow inhibit that protein, and that is how it's working. So we've figured out the mechanism of action of TMP by showing that all the bacteria that are resistant to TMP have mutations in this gene that change its shape, which strongly indicates TMP fits in. Now, you would actually want to follow it up with other experiments. Perhaps you mutate the DHFR gene to show that that alone can give you resistance to TMP. Maybe you start to do structure activity relationship experiments or computer modeling to show that the molecule actually fits in. And so TMP is now ineffective in the mutant model. And here's what they actually did. They followed it up with their computer modeling. So in black is your TMP antibiotic, and in gray is the protein, right? So there's the DHFR. It's actually not bad. It looks kind of like my Pac-Man. If you tilt its head, I drew that not too bad. The structure of the protein. And each of these colored dots is a mutation that occurred in the bacterial strains that conferred resistance. And you can see that all these amino acid changes from the DNA changes are all in that active site right around that TMP pocket. And so now we can totally understand how it's, by changing the amino acids, we've changed the shape of the pocket. And so now the drug can't fit into the pocket. Now, when I say shape, I do actually mean shape, because the physical shape, this is called, what is it called? Stoichiatric, no, I've forgotten the word. No, I'll pop it in the comments below. Can't get it. Anyway, there's physical shape, and then there is also a whole bunch of other interactions going on. So there is positive charges to negative charges. So that's electrostatic forces, polar, non-polar interactions, and hydrophobic stacking, for example. So if there's in that pocket a little hydrophobic region that doesn't like water, and the drug has a hydrophobic region, for example, these rings right here are going to be pretty hydrophobic, these carbon rings. And hydrophobic things like each other, so that will allow the drug to sit into that hydrophobic region. Now, if you swap out one of those amino acids from a non-polar amino acid to a polar amino acid, suddenly the hydrophobic interactions can't occur. So when I say shape, I kind of mean all the stoichiometry. I think that's the shape. Yeah, I kind of mean all of the chemical interactions that occur between a drug and its target, which isn't just shape, it's also charge and hydrophobicity and all that kind of stuff. Hydrogen bonding, all sorts of stuff happens at a drug site to a protein. But they actually did more than just one antibiotic. So here we can see some of the flaws of this technique. So TMP, it worked amazingly. Here's the TMP treated bacteria, and here's the gene, the promoter region of the DHFR gene, and the actual DHFR coding region. And then each of these shapes is a replicate, and they've put the shapes to correspond to a mutation. So all 4 of the 5 ends got a mutation here, 1 of the ends got a mutation here, 2 of the ends got a mutation here, so on and so forth. So you can see there's loads of mutations, and they're all in this DHFR gene and promoter region. And so it's all to do with folic acid synthesis and metabolism. But there were other mutations. So there was a couple of mutations over here and a couple of mutations over here. And these are different things. So these are membrane proteins and transcription and translation proteins. But for the TMP, it's clear that the vast majority of all mutations were in this one gene. But you can see some of these other antibiotics that they tried. So they tried doxycycline and chloramphenicol, or CHL. They tried these 2 other antibiotics. Now, so with doxycycline, it worked quite well. Doxycycline targets the ribosomes of bacteria. The ribosomes of bacteria are very different. They have different subunits, like the 16S subunit, compared to the eukaryote ribosome. So because of the difference in those ribosomes, a lot of antibacterials target ribosomes. So the doxycycline targets ribosomes, and what do we see? That's a ribosomal gene, that's a ribosomal gene, that's a ribosomal gene, and they all have mutations in the doxycycline. And then these are just regulatory genes of translation, right? So it's also to do with the ribosomal machinery, these other genes here. So what you can see is the majority of doxycycline mutations occurred where it works, which is at the ribosome. But there were some mutations here that occurred in membrane proteins, which is interesting. That's not where it works, but we found all these membrane proteins contain mutations for doxycycline, for the doxycycline-treated bacteria. And this was even worse for the CHL antibiotic, where almost all of the mutations were in the membrane proteins, whereas we know through other techniques that CHL actually works at the site of the ribosome. So there were a few mutations in the ribosomal proteins, which is what we'd expect, but actually the majority of the CHL mutations occurred at the membrane proteins. So what's going on? Why do we have weird mutations occurring in membrane proteins when that's not the drug target of those antibiotics? Well, one likely explanation is that bacteria have these pumps. Now, these pumps are designed to pump out molecules and pump in molecules to the bacteria. So they take molecules from within the cytosol and they whip them out of the cell, or they take molecules out of the cell and they whip them in. Now, sometimes the antibiotics use these pumps to get into the cell, and a lot of the time bacteria evolve pumps to get the antibiotic out, right? So these mutations could be to do with those 2 things. One, they could improve the bacterial pump that removes the antibiotic from the cytosol, just keeps pumping it out massively. That's a really common form of antibiotic resistance. So here we have a drug efflux pump that the bacteria have evolved. So the antibiotics are actively shuttled out of the cell so they can't work on the ribosome. So that could be one mechanism by which these mutations are allowing them to develop resistance to the CHL and the doxycycline. Another one is that these pumps can sometimes pump the drug in, and so you can mutate it so it doesn't allow the drug to get into the cell, right? So then we've learned something really important, which genes are associated with pumping the antibiotic in and out, but we haven't learned the mechanism. So CHL, from this experiment, if we didn't know how it worked, it would be very difficult to figure out how it works, because the majority of the mutations are in this membrane proteins, which might be a bit of a red herring about how it works. And we had a few mutations in the ribosome. So we might then explore those with specific mutants. So we might generate a bacteria that has these mutations in the ribosome and see if it's resistant, even without the mutations in the membrane proteins, for example. And then we can also do computer modeling or pull down experiments, which I explained in the previous video, or RNA sequencing, which I explained in the previous video as well. So then we'd have to explore our other avenues. But you can see it is a really good start. It helps to narrow down how your drug might be working. And sometimes it just straight up gives you the answer with this TMP example. But I love this method. The idea of using evolution to figure out how your drug works is so clever. And I know it's a little scary. You're now generating an antibiotic-resistant bacteria. The one thing I'll say about that is that mutations come at a cost, right? So for example, if we were to remove the TMP pressure, so now the bacteria are being growing in regular conditions, and we were to just grow them for say a month in regular conditions, what we might expect to happen is these mutations in the DHFR gene to be reversed. Why is that? Well, let me just pop back a sec. This shape here might be less effective at its folic acid metabolism, right? So you think the unmutated gene has evolved over billions of years to perfect this metabolism. It has evolved so perfectly to do this action, right? And any changes to that is likely to reduce its efficiency, but that reduced efficiency is worth it in the presence of an antibiotic. So in the presence of an antibiotic, it's worth being 20% less efficient in order to not have that antibiotic work on you at all. Right? So it's all about trading off the selective pressures. But if you remove the selective pressure by not having that drug anymore, you would expect the mutations to start to go back to the wild type version of the gene. And this is why antibiotic resistance doesn't become a sustained, often, it does sometimes, but often doesn't become a sustained mutation that's present in the soil bacteria, for example. And why we often find the largest populations of antibiotic-resistant bacteria are in the hospital, and that's because they're under the constant selective pressure of those antibiotics. So it's always worth having those mutations if the bacteria endemically grows in the hospital. But in the soil, it's probably not worth having those mutations, so sometimes those mutations will literally undo themselves. They'll flip back to the wild type. So it's not as dangerous as you think to generate an antibiotic-resistant bacteria, because those mutations may not be sustained if it ever got released into the wild, but you certainly wouldn't want to get it in you or on you. And sometimes what we do to make this research safe is we mutate these bacteria intentionally to lack certain virulence factors which are essential for infecting humans. Okay, so to make all this research safe, what we sometimes do is we take the bacteria and we mutate it so it's lacking a critical gene that's essential in it becoming an invasive bacteria in the human. For example, the fimbria, which are processes that go off the bacteria and allow it to stick and adhere to our cells. And sort of navigate our environment, avoid the immune system, for example, the fimbria, we might remove those fimbria. So now the bacteria can't do things like climb the glass wall on the flask, and they can't get in and attach to our intestine, for example. So we make them useless, but perfectly happy to be grown in a culture medium. But if they ever got into us, they would be useless. And that's another safety feature that we put into our research. We also certainly don't drink anything that we have in the lab or anything like that. Thank you very much for listening. I hope you found that one a good one. Pop any questions below in the comments and I'll be happy to answer them. Thanks, team.